Hello, I am Dr. Kristen Honey, the Chief Data Scientist in the Office of the Assistant Secretary for Health at the U.S. Department of Health and Human Services, or HHS. I'm also the co-founder of our LymeX partnership, our historic $25 million public-private partnership with the Stephen and Alexandra Cohen Foundation. First and foremost, I'd like to thank the community for supporting LymeX. Now, let me turn it over to our LymeX team to talk about our LymeX Innovation Accelerator.
Hello, my name is Dr. Daniel Desautel, and I am a fellow with the LymeX team. Our work at LymeX involves taking a human-centered approach to engage diverse voices across all sectors and disciplines. Through an interagency agreement with the NASA Center of Excellence for Collaborative Innovation, the LymeX partnership is now building upon almost 700 hours of human-centered design, workshops, interviews, and listening sessions to kick off our prize competition called the LymeX Diagnostics Prize. We expect this to be a multi-year, multi-phase competition to develop diagnostics that detect active infection by Lyme disease-causing bacteria in people to be submitted to the Food and Drug Administration for review. We recently announced the winners of the first phase of our LymeX Diagnostics Prize competition. Let's hear from the winning teams on what solutions they are developing.
Hello, I'm Dr. Mark Eshu and the founder of Blue Arc Biosciences. We are one of the recipients of the LymeX Phase 1 prize. So we've been working on Lyme disease diagnostics for over 15 years. One of the things we've learned is that you need to be exquisitely sensitive. With LymeX funding, Blue Arc Biosciences is developing ultra-sensitive PCR-based assays for the detection of early Lyme disease.
I'm Dr. Marianne Communell from Drexel University College of Medicine, Department of Microbiology and Immunology. We're developing a glycobiology-based diagnostic test that can be used to detect early acute Lyme disease infection prior to seroconversion. This approach is also being used for subsequent infections and to track disease state. Thanks to the HHS and Cohen Foundation for making Lyme be their 2022 challenge. We hope our diagnostic will alleviate suffering due to delayed treatments.
Hi, I'm Dr. Leota.
Alessandra Lucchini.
I'm Paul Russo. In a clinical study of Lyme disease patients, we identified hundreds of novel biomarkers shed into the urine by the Borrelia bacteria itself. This is a direct test.
Now we want to translate this experimental discovery into a standardized clinical test to reach wide public benefit. We're going to use a novel biomaterial urine collection device and new technology to bring the test to any clinical lab.
Helix Bind is developing an ultrasensitive molecular diagnostic for the direct detection of Lyme borreliosis. Our test combines novel sample preparation with a unique detection strategy to identify active infections in blood and other bodily fluids. This test aims to markedly expand the detection window, reducing diagnostic uncertainty and enabling the initiation of treatment when it is most effective.
Hi, I'm Aarti Vardur, and I'm part of the R&D group in Bios International Inc. We are in Seattle and make robust diagnostics for a variety of infectious diseases. We are very excited to announce the development of a novel tool to diagnose Lyme disease. This has a microarray immunoassay with a wide variety of Lyme targets and a machine learning algorithm to help accurately determine a patient's status. This will vastly improve the sensitivity and confidence in detecting early Lyme disease.
Hi, I'm John Branda. I'm a pathologist at Mass General Hospital and Harvard Medical School.
And hi, I'm Jake Lemieux. I'm an infectious disease specialist at Mass General Hospital and Harvard Medical School. Our team's goal is to use plasma metagenomic sequencing and target enrichment to identify Borrelia burgdorferi from patients with Lyme disease.
Based on our preliminary studies, this novel approach has the potential to increase the sensitivity and specificity of existing diagnostic approaches. Our overall goal is to develop an accurate test for Lyme disease during all stages of the illness.
Hi, I'm John Son, the Chief Technology Officer of Seramune, a biotech company based in Santa Barbara, California. We offer a technology called Serum Epitope Repertoire Analysis, or SERA, a transformative precision serology assay that uses large data sets and machine learning to develop best-in-class diagnostics. Using SERA, We identify antibody epitopes in each person with a patient-oriented, home-based sample collection kit to detect and diagnose Lyme and other tick-borne diseases.
T2 Biosystems was the first company to receive FDA clearance for detecting sepsis-causing bacteria directly in whole blood. Now we're applying our proven T2 magnetic resonance detection method to Lyme disease. Currently, there are no sensitive FDA-cleared diagnostics for the detection of early Lyme disease, and we've developed a test to detect active Borrelia infection, the bacteria that causes Lyme disease. We believe the T2 Lyme panel will allow clinicians to accurately diagnose Lyme disease and ensure patients receive appropriate therapy faster.
Hey, I'm Pete. I'm leading the team at Tufts University in Boston. When people get treated for Lyme disease, most of the time they get better, but sometimes they don't. At the moment, we don't have a good way of measuring that. So what we're working on with the help of LymeX is a new test that we can use to track people during treatment and after treatment. So if the treatment works, you've cleared the infection, you feel better, great, we can give you the all clear. If the treatment hasn't worked, then we know and we'll be able to measure that, so we'll be able to make sure that the people who need it get the right treatment and get the right amount of treatment.
My name is Brandon Jutras. I'm an assistant professor in the Department of Biochemistry in the College of Agriculture and Life Sciences here at Virginia Tech. And I'm the team leader, but we have two other fantastic team members. One is Dr. Rich Helm. He's an associate professor also in the Department of Biochemistry, but he's also the director of user facilities for the NSF Material Innovations Platform. And our final team member is Marcos Pyers. He's a chemist at the University of Virginia. And what we are working on is a biomarker for Lyme disease diagnostic tests. And so basically what we found is when the bacterium that causes Lyme disease is transmitted from a tick into a human host, it starts to replicate. And as it replicates, it sheds its cell wall, or specifically a molecule called peptidoglycan. Now, this is a very abundant molecule that's naturally being shed by the bacterium. And most importantly, this molecule is extremely unique to the bacterium that causes Lyme disease. And so what we're developing is a sophisticated but very accessible test that can directly detect this molecule and, in essence, be able to diagnose Lyme disease within hours after infection.
Congratulations to the winners of Phase 1 of the LymeX Diagnostics Prize competition. Next, we'll hear from another LymeX team member, Kathleen Carroll, who will discuss our commitment to centering your voices.
Hello, my name is Kathleen Carroll, and I am a U.S. Digital Core Fellow with the Office of the Assistant Secretary for Health. We at LymeX are committed to including the patient voice in all we do. Through Health Plus, LymeX is listening and learning from individuals living with Lyme disease and the latest scientific guidance to facilitate patient-centered innovations and improve health outcomes. Co-creation is at the core of what we do. Your voice, your collective action, and determination have been the cornerstone that we here at HHS have been building from to drive transformative change together. To stay informed and participate in upcoming LymeX workshops, listening sessions, and events, you can follow us on Twitter at Lyme_X. Your involvement, lived experiences, and partnerships bring us one step closer to our ultimate goal, for vector-borne disease to no longer threaten human health and well-being.
Thank you all and the LymeX community for being a part and for bringing about the Lyme tipping point.